Hello everyone, today I'll be talking about our project on the characterization of Arthobacter globiformis bacteriophages Crewmate, Lizalica, Cassia, ObiToo, Warda, and JohnDoe using phylogeny.fr and gene content similarity programs. So let's start with some background as to where this project came from and what we're doing. So 6 AZ cluster bacteriophages were isolated at the University of Ottawa as an extension of the C-phages program. Phages were isolated using Arthobacter globiformis, or B2979, as a form of the host bacteria. And as we can see from the TEM images, all 6 phages belong to the Fawagreh family, as they all had the characteristic tail and the icosahedral capsid head. Our work involved further characterization using 2 comparative genomic softwares, phylogeny.fr, which generates phylogenetic trees by comparing amino acid sequence of specific proteins produced by different phages, and the PhagesDB embedded gene content similarity software, which compares the GCSs of different phages, calculated by the shared number of FAMs from each phage and the average of the value for both phages. Our main goal of this project was to further characterize these 6 selected AZ cluster bacteriophages using those 2 softwares, phylogeny.fr and GCS, to look for key similarities and difference that are unique to this cluster of phages. So just a brief overview and a reminder of our methodology. We collected soil samples from local areas in Ottawa and went through a long procedure that allowed us to isolate, amplify, characterize, and purify the phages. Just to reach the most important part, the My Sequencing Library gene sequencing done at the University of Pittsburgh. And following this step was the genome annotation step, where we use different softwares to annotate and compare the phage genomes via different software such as the Pecan, FamWriter, GeneMark, HHPrad, and the different BLAST softwares. And our project focused on the phylogeny.fr and the GCS software to gather our data. So these are the results obtained from phylogeny.fr. And as you can see, for each gene of interest that was selected, a phylogenetic tree was generated and produced, and we can see all 6 phages present. So each branching event is essentially the introduction of a new evolutionary difference between the phages. For example, for the terminase small subunit protein, right away we see that Lizalica branches off just from the rest. We then have further branching, and we see that JohnDoe and Warda are on the same branch, and this branding pattern and these differences indicate that the terminase small subunit protein in Lizalica is likely much different than the remaining 5 phages, but for JohnDoe and Warda, this protein seems to be quite similar. And we can see the same thing happens for the terminase large subunit. Cassia is quite different from the rest as the branching event occurs right away. These are other values and other phylogenetic trees for the other selected genes of interest. But as we see here, these red values are bootstrap values, and higher bootstrap values indicate more confidence in that branch. So there's more likelihood that this evolutionary event did in fact happen. So for example, in the HNase endonuclease protein, we have a bootstrap value here of 0.043. This indicates that there was only a 4.3% chance that this branching event did actually occur. So it is quite likely that Warda isn't actually different from the remaining 6 phages in terms of its HNase endonuclease. We can see here, however, in the serine integrase protein that the bootstrap values are quite high, and these branching events likely did occur. So here we have a bootstrap value of 0.99. So it is likely that Crewmate and ObiToo are quite similar to one another, but different from the rest. And there's a 99% chance that this branching event actually did occur, and that they are quite evolutionary different from the rest. Here's the data from the GCS software. So essentially we can correlate the phages that were least similar from the results we saw in the phylogeny trees and consolidate them with the results from GCS. So for example, Lizalica has pretty low scores for most phages, except it is most similar to Warda. ObiToo and Crewmate are also quite similar and close to each other. Again, Warda seems to be the most similar to Lizalica, as we mentioned, and potentially JohnDoe as well. And each phage seems to have other phages that they're quite similar to, or they're quite similar to all of them. The only odd one out is Cassia, as it isn't extremely similar to any of the other phages, as the GCS content or the GCS value is lower than the rest. In conclusion, based on the patterns observed in the phylogenetic trees and in the data obtained from the GCS profiles for each phase, within these AG phage clusters, there's definitely some phages that are closer to one another than others, and there are definitely some phages that aren't close to any other phage at all. So generally, we saw that Warda, JohnDoe, and Lizalica are quite similar to each other in terms of their GCS and their phylogeny values. Crewmate and ObiToo also often seem quite similar to each other based on the GCS values and the phylogenetic trees. And Cassia also seems to be the least similar and had the lowest comparable GCS values to each other phage. In terms of phylogeny, the genes that are closer to the start of the phage genomes are more similar and conserved within the phages, while the genes closer to the end of the phage genomes are less similar. And this is something that we noticed based on the Famarator reports and the phylogenetic trees values that we obtained from our data. In terms of future directions, there are many things we can do. We can use these comparative genomic softwares to compare AZ cluster phages with other phage clusters, such as EA and FM, which we know that the University of Ottawa is currently working on as well. We can use different comparative genomic softwares to consolidate the data obtained from phylogeny.fr and GCS. And we can evaluate the GC content, which is essentially the percentage of guanine and cytosine in the phage genome, to help determine if any genes were actually acquired from horizontal gene transfer, which is the transfer of DNA between genomes of different organisms. So from a phage to another organism to a phage. And finally, we'd like to acknowledge everyone that helped us, and this project was made possible due to the support of Dr. Adam Rudner, who was our principal investigator, and Malizia, our lab TA, as well as the rest of the professors and TAs involved in TMM 3300 this semester, the University of Ottawa Faculty of Medicine, the C-Phages program, and finally, everyone at the HHMI. Thank you.